Hello everyone, this is Sana Parveen here and welcome back to my channel Sana's Biochemistry. In this video, we are going to talk about molecular kiss of death. Actually, when I heard this, I too myself was intrigued that what is this topic? So I thought of making a video on it. Let us just dive into it. Molecular kiss of death, actually this topic was for the Nobel Prize in 2004 in the field of chemistry. Nobel Prize was awarded to three different scientists named as Irwin Ciechanover, Avram Hershko, and Irwin Rose. These three researchers gave us the mechanism behind molecular kiss of death, and these were awarded Nobel Prize in 2004 in the field of chemistry. What did these scientists say? They were finding out the protein destruction in our body. How is the protein getting destructed in our body? Okay, not the proteins which are useful are going to be destructed by the body. The proteins which are useless are just taken care of by the body, because of which the mechanism was found out by these three amazing scientists, and then they were awarded the Nobel Prize for their work. Okay, Ciechanover, who was an Israeli biologist, gave us a statement and said that we human beings are not a building that stays still. He is comparing human beings to what? A building. So we are not a building, because building is going to stay, still forever. We are not like that. The building is made up of the building block called as a brick. Okay, similarly, our body is made up of various kinds of proteins, and the building block of protein is called as amino acid. Actually, I'm trying to make a link to proteins and the molecular kiss of death and the mechanism that these scientists gave. We humans are not still, that stays, we're not like a building that is going to stay still. We are all the time exchanging our proteins, synthesizing and destroying them. Some proteins also get spoiled. In our body, there are various proteins, as I just said. These proteins are getting exchanged in different forms. For example, this is the shape of the protein. This is one kind of protein. I don't want the protein to be in this shape. I just changed the shape of the protein. Okay, the body has got the capacity of doing that. Body can also synthesize new proteins. How is the body going to synthesize new proteins? By joining various amino acids into it. They can also destroy some proteins. How is the destruction going to happen? The long chain of amino acid, which is nothing but the protein, are going to be converted into small units called as amino acids. So body has got the capacity of synthesizing as well as destroying the proteins, and the proteins sometimes get spoiled as well. For example, this is a protein. Okay, the protein is now young and all good to do work. With the time, what happens is the protein starts to grow old. When this protein is growing old, the capacity of working of the protein decreases with time, and then the body is not in need of this protein, and then this protein is taken care of, is killed by the body. Okay, we discovered the process by which the body exercises quality control. The quality of the body is to be maintained all the time. So this was discovered by these three scientists, and this was told to an Israeli newspaper by Ciechanover. Okay, so the scientist, the main scientist, the leading scientist gave us a statement about this. Out of the three scientists that we were having in the discussion, who were awarded Nobel Prize altogether for their work on the mechanism of destruction of proteins, out of which Ciechanover gave us the statement, which is like having a very direct link to molecular kiss of death. We're just approaching there. Okay. Yeah, Ciechanover looks handsome. I just saw him. Oh my god. Yeah, let us move on. Okay, I'm not able to get over Ciechanover, but yeah, let us continue with the video. Okay, so here in our body there are some proteins called as non-functional proteins, denatured proteins, and defective proteins. The protein is there, but it is not ready to function. The protein is there, but it is in the denatured form. The protein is again there, but it is in the defective form. What is the meaning of denatured protein? For example, this is a protein, and because of temperature and pH changes, or a high concentration of salt, The structure of the protein is changed, is open up, and the protein is called to be denatured. For example, when you get an egg and you put it on a high temperature pan, the pan is really hot, the protein gets denatured and converts from the liquid part of the egg to the solid omelette part. Okay, so that is called as denaturation. The same thing happens in curd as well. Okay, we have got the curd, we make it from the milk, and then the curd is having a different appearance than the milk. The milk was like very liquid, very fluidy, but the curd is semi solid. That is all the examples of denatured protein. In our body, when we have got the non-functional, denatured and defective proteins, our body is not in need of these proteins, and these proteins are just taken care of and is just thrown out the body, or like recycled, or whatever the body wants to do out of it. Next thing, kiss of death. So we are, moving into the topic, and what is it exactly? The kiss of death for protein is called as ubiquitin. Who is going to cause, who is going to help in killing the proteins is the ubiquitin molecule. This ubiquitin molecule is itself a protein. Okay, this is a protein and is going to cause the death of other proteins. Okay, not having pity on them. It is made up of 76 amino acid and it causes the kiss of death to the other protein molecules. This is done by tagging proteins with ubiquitin and is called as ubiquitination. So this ubiquitin is ubiquitous. That means it is present in most of the cells and tissues in eukaryotic organism. Okay, so ubiquitin is present ubiquitously and is involved into a process of ubiquitination, which is called as tagging the proteins. Which proteins are being tagged here? The proteins which are useless, the denatured protein, the non-functional protein. Okay, so these proteins are going to be tagged by the ubiquitin molecule. We're just moving ahead of that. Okay, now consider I have got a building. Okay, just let me take a color which is like so visible to you people. So this is the protein and this is a building. I want to destroy this building, okay, because the building was illegal. I call a JCB, a crane. There are many buildings in that area, there are many buildings in that area, but I need to tell the person which building is to be broken down, right? Otherwise the crane is going to come and just destroy all the buildings. We don't want that. We want one specific building to be destroyed. So the crane person will be given instruction. Okay, so the second building, okay, I just put I'm just going to put a red color kind of a thing. I'll just write on it saying destroy. When I write on it destroy, you just have to destroy that protein. Okay, so let us just move ahead now. So ubiquitination, now you'll understand a bit clearly when I show you the diagram. Okay, now what is happening here is ubiquitin is going and tagging the proteins which are to be destroyed. Okay, so the non-functional proteins are to be destroyed. Ubiquitin is going to go and kiss that protein. Okay, kiss of death. The death is going to happen once the ubiquitin comes and kisses that protein. Okay, be very careful about this. So the kiss here is lethal. It is going to cause the death of the protein. Oh my god. Yeah, so it is the ubiquitin will come and kiss the protein, the non-functional protein, the denatured protein, and the defective protein. The case here is it can happen that one ubiquitin is coming and kissing, or sometimes two ubiquitin will come and kiss the same protein. Sometimes what happens, many ubiquitin molecule are going to come and kiss the protein which is to be destroyed, and this process is called as ubiquitination, and this process is also called as tagging the protein for its destruction to happen. Okay, once the protein is tagged, let us just move on. Once the protein is tagged, see, you can see the tags present in here. You can see the tags present in here, 1, 2 and 3. Three ubiquitin molecules are present here. So there are many ubiquitins labeled to or tagging just one single protein. Okay, or it can also happen that only one ubiquitin is sufficient enough to cause the destruction. Ubiquitin is the signal, gives the signal to the cell transport machinery to take care of the protein with the help of another destruction molecule which is going to cause the destruction, is called as proteasome. Okay, so the crane is going to have a person in it. Okay, so the crane is operated by the person. The crane, this is the crane. Okay, ubiquitin is the tag who's going to help in the destruction. So I am proteasome, I have destructive mentality. Oh my god, the proteasome has got destructive mentality, and I am here to kill that protein. Which protein? The labeled protein. Okay, so this, what is the protein saying? Someone please help me. Proteasome is going to kill me as ubiquitin labeled me. So it's a teamwork by ubiquitin and proteasome to take care of the destructive protein. At the end, what happens? This proteasome has taken care of the destructive protein, of the useless protein, and causes the destruction and breaks into smaller pieces. And sometimes what happens is these smaller pieces are just, are not needed by the body anymore, or just can be thrown out of the body. Or sometimes what happens is these particles, these parts of the proteins, are used as raw material for synthesis of another protein. These ubiquitin molecules come other side, and they can be used for, for repeating the cycle once again. So who is causing the destruction? Proteasome. What is molecular kiss of death? Let us just, conclude with it. Molecular kiss of death is when you have got a defective protein. That defective protein is kissed by the ubiquitin, so that the defective protein is labeled for its destruction. The proteins which are useful are not labeled by ubiquitin. The ubiquitin is not coming and kissing the useful protein. It is coming and kissing only the useless protein for its destruction. So after labeling, after ubiquitination, there comes a proteasome which is going to kill the protein into smaller pieces. So the last point in here says that ubiquitin system is the subject of pharmaceutical research. Drugs could be developed to prevent degradation of specific proteins, or to trigger the system to destroy unwanted proteins. So the drugs can be designed to prevent degradation of specific proteins. So for example, some proteins, I don't want the destruction of these proteins to be happening by the labeling, by the ubiquitination process. So drugs can be, made for doing the needful. Or what you can do is trigger the system to destroy the unwanted protein. All these things can be taken care of. And then, yeah, so thank you for watching the video. Actually, this was the seventh time that I recorded the video and possibly happened. So if you find any, fluctuations in the video, kindly ignore that. I hope that the information is useful to you people. And if you like the video, hit the like button, subscribe to my channel, hit the bell icon as well, so that you'll be notified for my upcoming videos. By that time, stay safe, stay at your home, because corona time. Okay, so everyone need to take care of themselves, their loved ones, the old people, the younger kids, everyone. So stay at home, plank the curve, take care of yourselves, hit the like button, don't forget that. And yeah, take care, bye bye, see you in the next video.